Hi, I'm Jorge. I'm the leader of the Science Communication Committee, and you're all waiting for the speakers to talk. You don't want to listen to us anymore. So I'll introduce the first speaker very quickly. We have the honor of having here today Professor Carmen Torres Manrique. He's a leading expert in clinical microbiology and antimicrobial resistance. She's a professor at the University of La Rioja, where she heads a research group studying how resistance spreads across humans, animals, food, and the environment. Her team's work has gained international recognition, including the 2022 Spanish National Award for Initiatives Against Antimicrobial Resistance. Carmen, it's a real pleasure to have you here. Thank you for coming, and the floor is yours.
Good afternoon to everybody. It's an honor and a responsibility to have been invited to participate in this Spanish influencer in science session. Thank you. I would like, first of all, to thank to CEBE for inviting me to this event. And also, I appreciate to be in this marvelous place in Institute of Cervantes that for all Spaniards is a very, very important place. And of course, the Embassy of Spain and the Fundación Ramón Areces that also supported this action. So we are going to talk today about a problem that affects to all of us, and I am going to do it from the One Health perspective, antibiotic resistance from the One Health perspective. Thinking in what I was going to say, I thought that there was in 1988 an important event. It appeared, it emerged, a type of resistance, a vancomycin-resistant enterococci in hospital in that moment in France and in United Kingdom, but not outside hospital. And 3 decades ago, we published the first paper in which we detected this resistance in sewage, in enterococci that carry this specific resistance. Later, we knew that this resistance came from the use of antibiotic in animals as a growth promoter. So it was 3 decades ago, just the beginning of the concept, One Health concept related with antimicrobial resistance that join hospital, the environment, and the animal. 3 decades, we are now with a problem, a real problem of antimicrobial resistance. Health and scientific authorities are very, very worried about okay. It's okay? Yeah. Okay, better now. Health and scientific authorities, political and economic authorities alert about this problem, and the World Health Organization consider antibiotic resistance as a real problem, as is one of the highest public health challenges that could put us in the post-antibiotic era. And the data. We know a lot of data about the death that is caused by antimicrobial resistance. If we use this paper that is very recent and refer to the number of deaths related with antimicrobial resistance in 2019 at global level, directly attributed, we can found 1.3 million and associated 4.5 million. It's a lot, and the forecast for 2050 is very, very unhappy for us if we don't take the measure and the control just to reverse the situation. And if we analyze the type of bacteria that are mostly causing this death, Escherichia coli, that I am going to talk later, is the first one. we know that antimicrobial resistance is the evolutive answer, response to the intensive and continuous use of antibiotic in humans, in animals, and in a lesser extent in agriculture. We use for treatment, we use for prophylaxis, and for a long time have been used as growth promoter. But in the European Union is already banning the use of antibiotic as growth promotion, but is still used with this purpose in many other extracommunitary countries. And what do antibiotics when we use? When we use one antibiotic, it affects to the bacteria that we are treating, the pathogenic bacteria, but as a secondary effect, is also affecting all the microbiota that is very, very rich that we have, and not only we, also the animal, and can emerge mechanisms of resistance, for example, in the intestinal microbiota. The bacteria, through the feces, goes to the environment, can contaminate the environment, can contaminate the food, and in this way, bacteria and genes of resistance can spread in the different ecosystem. In this way, we have also to take into account that bacteria are very old, are the oldest organisms in the earth. So they have learned very well how to adapt to the antibiotic, and they have a lot of platform and a lot of strategy to become resistant. We have integron, we have transposon, we have plasmid that can carry 1, 2, or 3 or more genes of resistance. And in this way, the acquisition of this element can produce a multi-drug resistance, could be co-selected by the use of different compounds, and not only antibiotic, also biocide, metal, because resistant gene for all these compounds can be integrated in the same structure. In addition, bacteria can interchange genetic material, and this interchange happens when in the intestine they are very close and they can interchange the plasmid of resistance. So a bacteria that is susceptible, that is the one that is in the left, can become completely resistant, and it's not possible to use antibiotic to treat them. That is the one to the right, and it can be even multi-resistant. So the way to manage and to see nowadays the problem of antibiotic resistance, we have to look in the One Health perspective. One Health perspective because the health of the human, the health of the animal, and the environment are completely interconnected. So what we do, the humans, what we do with the animal affect to all of us. And we have to go ahead because it's not only One Health. We have to talk of the global health. We know that there are conflicts, for example, the war, that are a very driving force in the dissemination of antimicrobial resistance, meteorological disaster, travel, international travel, animal migration. We are going to talk about this later, and the trade of animal and food that can move all bacteria. The bacteria do not have border, so they can be transmitted in the different ecosystems. All the countries, or many countries, has developed a specific action in order to fight against antimicrobial resistance. In Spain, we have the Plan for Antimicrobial Resistance, that is the PRAN, that started in 2014, and we have nowadays 4 specific projects. The actual one is from 2025-2027, that is completely multidisciplinary, multisectorial, and transversal, and participate all the communities and many researchers and community and scientific communities in order to get the way to manage the situation of antimicrobial resistance. We have We have the data related with 2014-2021. Spain, we have been always in the top of antimicrobial use and antimicrobial resistance. We are happy because with this action, we got a decrease, important decrease in the consumption of antibiotic in human and in animal. We have to continue because we still are with a lot of consumption, but things can be done and things are done through this national plan. And the environment is the forgotten ecosystem. We know what is happening in the human, we know what is happening in the animal, but the environment is the forgotten ecosystem, but antibiotic, antimicrobial resistant bacteria, and antimicrobial resistant gene arrive to this ecosystem. So it's important to have also a specific action for the environmental sector. In this sense, the PRAN, this national plan, has produced two documents in which I have had the opportunity to participate in the work group to produce the document in which the source is are analyzed, the sources, dispersion route, objective, strategies. So it's the first step just to go to this environmental sector. And what are we doing in the University of La Rioja? I am from a group in the University of La Rioja that is antimicrobial resistant from the One Health perspective, OneHealth-R, that I coordinate, and I have very nice scientific that we work trying to study commensal bacteria that can be also important opportunistic pathogen, and we analyze in healthy and sick humans, in food-producing animals, companion and wild animal, food and the environment. And what do? We analyze the mechanism of resistance, how these bacteria become resistant, how they transfer the gene of resistance between them. We do the molecular epidemiology just to see how they move, and we do very, very profound genetic analysis. We analyze the evolutive marker and how bacteria can move from one host to the other, and we try to look for new strategies, new antimicrobial. We are a multidisciplinary group. In our group in the University of La Rioja, we have people with many different expertise, because if we want to work in One Health, One World, we need people of different research with different expertise. And we collaborate with research groups from many places in the world. I have put several points, and we try in a collaborative way to work with countries and with research groups that have different policies of use of antibiotics, environmental laws, resources, good food habits, and human-animal relationship. We work always with bacteria that are commensal, that are our own bacteria, but can become important pathogen, because these are very important to track the evolution of resistance in these bacteria. Our work is in public health, analyzing the clinical interest, food security, and the veterinary interest. Also using these bacteria, Escherichia coli, Enterococcus, Staphylococcus aureus, as sentinel of antimicrobial resistance, because we know that are the most relevant opportunistic pathogen. I am going to put an example of the One Health perspective, taking into account one specific resistance, that is the production of extended spectrum beta-lactamases and carbapenemases in E. coli. E. coli is this bacteria that is the most relevant causing death in global health, but that is a bacteria that we have in our intestine. Why we have selected this specific bacteria and these specific resistances? because the World Health Organization, they consider critical group. And to be considered a critical group is infection with limited treatment option due to resistance to last resort antibiotic. And these are these beta-lactam. And the ability to spread across different ecosystems. E. coli is perfect, is our friend that we have in our intestine, but at the same time is the most relevant opportunistic pathogen. Considering the resistances, we know that before the 90s, it appeared different report, but very specifically related to Klebsiella pneumoniae, to hospital setting, and to variants that were different to the one that I am going to talk about, that is CTXM. But in 2000 happened an explosion. Explosion of what? Explosion of E. coli harboring this type of beta-lactamase. Initially in hospital, in the community, was the beginning of the, what we say, CTXM history within the One Health framework. This type of resistance that appeared at the beginning of the 2000, we see in color how the different countries were becoming more brown, even red, that indicate how were evolving this type of resistance. Also, healthy humans were becoming colonized by this E. coli carrying this mechanism of resistance. And importantly, there are an important difference depending on the areas considered by the WHO, the World Health Organization. The areas, the poor areas, the areas with low income were much more colonized by this type of E. coli producing this type of beta-lactamase. There were specific bacteria that become epidemic. This CTXM is a mechanism that obtains the perfect host to disseminate. And it's CTXM15 in a specific host that is E. coli ST131 that spread very easily in the different hospitals in all over the world. But what we have done in this area? What is happening in animal? We know that in human, the explosion was in 2000. But in 2000, in the early 2000, our group, in collaboration with other groups, presented the first report of this type of resistance in pets, in food-producing animal, and in wild animal. These are very important milestones of our group in this specific topic. We have performed in this area different collaborative studies in the Iberian Peninsula, in wild and free-ranging animal, food-producing animal, food of animal origin, companion animal, to know how this type of resistances were spread in this specific animal. And this study had been performed in other countries in collaboration with other groups that are indicated in the slide, in Mexico, in Chile, in Algeria, Nigeria, and Tunisia. But also in this environmental perspective, our interest was not only the wild animal or the animal, but also the water, seawater, the wastewater, and the superficial water, because it's a hot spot in the dissemination of antimicrobial resistance. Now there is believe that the epidemiology of wastewater is very, very relevant. With the COVID, we saw the relevance of wastewater to follow the different variants of the COVID, but also in antimicrobial resistance is a very specific area. In the influence, the water that arrive to the wastewater treatment plant, they can give us information about resistant bacteria and resistant genes that are circulating in a population. We can even detect emerging mechanisms of resistance. And the effluent that arrive to the river can give you the environmental impact of this water and the efficacy of the treatment. So we have performed different studies in wastewater treatment plant, applying different technologies in collaboration with different groups, culturomic, qPCR, and metagenomic, to know how these mechanisms of resistance are spread out. And the same in water, superficial water and irrigation water that arrive to the farm have been performed by our group in collaboration with other groups in Mexico, Chile, Algeria, and Tunisia. Taking all these studies together, we can say that not only human clinical isolate in humans or in healthy humans that colonize this type of resistance is spread out, but also we detected a high prevalence in food-producing animals. The food chain is a transmission route of this type. Of this type of resistance, and higher prevalence in human-impacted wildlife. And wastewater is a critical reservoir and transmission route for all this. New strategies for to look for antimicrobial resistance. We can use different strategies that we can focus just to analyze very deeply the isolate obtained in different environments, just to know and to compare what with what we have in databases. So, for example, we can use broad genomic analysis to see, for example, if migratory bird can contribute to the spread of antimicrobial resistance. We can see, for example, stores that are fed in natural areas or in landfill, the effect of the food in this aspect, and also if the bacteria that carry the store can be similar to the genome of other bacteria that are along the migratory route. So with very deep genomic analysis, we can analyze, we can see the emergence in this type of animal of a specific resistance that are of interest. And in this study, we analyze more than 10,000 genomes of databases compared with our genome to see the similarities of the bacteria that carry the store with those genomes that are in the migratory route, to see similarities and to understand how migratory bird can also contribute to the transmission and dissemination of antimicrobial resistance. Even we have analyzed the similarity of some of these strains carrying these resistances with those of the hospital that was nearby, detecting similar characteristics at the genomic level. So wildlife is an important point. We can consider wildlife in the dissemination of antimicrobial resistance, and depending on the animal, we can look in short or long distances, depending on the model we use. So a stork may be contaminated by bacteria that could be of human origin, but can be non-human reservoir. The prevalence vary depending on the level of anthropization of a store population, and could be good sentinel in all this process. So antimicrobial resistance is a global crisis, and the One Health is the solution. We have to work together, all the sector, human, animal, food, and environment, to get a good knowledge of the situation. Without action today, no cure tomorrow. We need research, innovation, surveillance, engagement, new alternatives, as Professor Prof is going to explain later. So we have to take into account all this. And I would like to thank to my group in the University of La Rioja, and also to all the group with which we have collaborated along the year in many studies that we have performed in this area. But a special thanks to my maestro, Professor Fernando Vaquero. He awakened in me the passion for science and the admiration for bacteria. He was and will always be my influencer in science. So what I try is to transmit the passion that I received from him. He was my PhD advisor, and he has been always an inspiration for me. I hope that I could be in the future also inspiration for others. Thanks a lot.
Okay, let's go for our second speaker. And indeed, let's see if we can find some alternatives. Our second speaker is Professor Rob Levine. He's a professor at KU Leuven, and he's the head of the Laboratory of Gene Technology. And his pioneering research has led to innovative approaches like artilysins, and those are enzymes that can kill resistant bacteria. He also plays a key role internationally as president of the Society of Viruses of Microorganisms and co-founder of PHAGE, which promotes phage therapy. Rob, pleasure to have you here. The floor is yours.
Thank you also from my side. Thank you for the invitation. I have no Spanish roots. My research is on phages, but I do have one scientific hero who is Spanish, and that's Margarita Salas, who had spent her career working on phage. I've had the fortune of, in her autumn days, to collaborate and publish together. I hear from the reaction that most of you know her. If you don't, I think you have succeeded as a scientist if you get put on a stamp, right? I'm going to do the disservice to summarize her research in a single slide. So she studied from bacteriophages, these are viruses, and discovered during studying of replication and transcription an interesting enzyme called Phi29 that is very prolific in amplifying DNA. And you can say this is a very simple invention, and I've finished it in this small cartoon, but it spurred an immense fields in terms of cancer diagnostics, in terms of archaeology research, forensic research, anything where you have a small amount of DNA and you need to amplify it. So her discovery on fundamental, which is based on fundamental research, has had a lot of practical applications, and she has published over 300 publications. And one of her quotes is that fundamental research is important, and that we can learn unanticipated results from that. And I think this is a very nice example. As I mentioned, she studied viruses, replication, so do I. When we think about viruses, the one on the right needs no introduction. We have the COVID virus. The one on the left may look a little bit less familiar, but I'm here to tell you that there is also good viruses. And so while COVID is adept at recognizing and infecting human cells, we also have our bacteriophages that are going to recognize and infect bacterial cells. And we learned from the previous talk that pathogenic bacteria are our enemy. And this is the enemy of our enemy that makes us a very big, friendly virus to work with. So how do bacteriophages work? What do they do? Well, they very specifically recognize the bacterial cell, as you see on the left. They attach, because this is all a protein structure that you see. All proteins, I don't know if there are Lego fans here, but if you were imagined to take a box of Legos, shake it, and then it's all formed, that's basically what happens with these phages. All of these proteins you see, they self-assemble into this superstructure that is a virus. And this assembly leads to this infectious particle. And on the outside, you have this protein that serve as a sort of syringe. And in this way, the bacteriophage will inject its genetic material, its DNA, in most cases for phages, inject its DNA into the bacterial cell. And then within a matter of minutes, it's able to convert the entire bacterial metabolism into a virus-producing machine. So as a matter of minutes, all metabolism of the bacterial cell is stopped and completely converted and repurposed to create new viral DNA and new phage DNA, to create new phage particles like the Lego box, and then it has enzymes to degrade the cell wall, and the cell bursts open and the progeny phage are set free. Yeah? So this is very interesting. I think you will see later on. Two elements are critically important. One is these bacteriophages are incredibly specific, right? We have a lot of diversity of bacteria, while phages are going to specifically recognize one bacterial species or even subspecies. Yeah. On the other hand, one phage particle infects a bacterial cell, and you have dozens to hundreds of new phage particles that are being formed. So it's a self-replicating entity that if it reaches a site of infection, if it reaches some bacteria, it's going to self-amplify. And this is very unique. Yeah. So how does that look like? The phages were found over 100 years ago by a British soldier scientist. He created these agar plates and filled them with bacteria, a bacterial lawn. And at the site where there was one phage, it started amplifying and created this negative colony where bacteria could not grow because these phages were amplifying and killing all the bacteria in their path. And that creates these colonies. And it's a virus, but it's being called a bacteriophage from the Greek, bacteria eater. And in this way, in our research, we're trying to see very different ways on how can we repurpose bacteriophages, what can we learn from bacteriophages in the AMR crisis to see is there new antibacterial design strategies that we will get from it. But I told you we have to start with fundamental research. This is not space. The big dots you see are actually, this is actually a droplet of water from the ocean. The DNA has been stained, and the big dots you see are bacteria, and the small dots you see are actually bacteriophages that are present. So in our global ecosystem, bacteriophages outnumber bacteria 10 to 1. There is 10 to the 31st bacteriophages that are present in our oceans everywhere, basically. And that means that bacteriophages on a daily basis are killing between 20 to 50% of all bacteria, all keeping things in this very nice dynamic balance that keeps bacteria in check, but they don't kill all of them. So they have this dynamic of interacting with bacteria that also make sure that they still have substrate, that they still have bacteria that are present for them. So this is happening in the oceans, but this is also happening in your guts, as was very clearly indicated. Also a very complex environment of bacteria, a lot of healthy bacteria that are present, but also phages are there. So also this struggle between bacteria and bacteriophages is happening inside your body. But as Carmen clearly mentioned, if the wrong bacteria is present, yeah, so the non-commensal, we can get infections. And I couldn't have had a better introduction before. So these bacteria often carrying antibiotic resistance genes that may be emerging in our hospital and clinics. As was indicated, they can enter our sewage and water supply into the environment, to the animals, creating the One Health loop that was so clearly explained before. And in this loop, bacteria are exchanging their genetic material through plasmids and so forth. We are seeing similar scary things that are happening. And unfortunately now, this is one of the samples from Kiev that we've analyzed. So it was mentioned that war can be another driver, and indeed soldiers coming into the clinic and the isolates that we are analyzing from this are incredibly scary. You see here, this is Klebsiella pneumoniae. I don't know if you see it, but with this string, you can pull on the colony and it has the consistency of chewing gum. And this is how this bacteria protects itself. In addition, it's becoming omni-resistant to all antibiotics due to plasmids that Carmen explained that carry virulence, that carry antibiotic resistance gene. And these are other drivers that are compounding and making the problem worse. So in our lab, we try to harvest or be inspired by phages to develop new antibacterial design strategies. I only have time to explain one, but in general, we look at various mechanisms. One is this host takeover mechanism, the molecules that the phage is making to block the bacterial metabolism. Yeah, that's basically showing us the way through billions of years of evolution, how we can block the bacterial metabolism, which is exactly what a small molecule antibiotic also does. So maybe we can be inspired to create small molecules. Second, the enzymes used to degrade the bacterial cell wall and release those, if we take those proteins, apply them to bacteria from the outside, can we destroy the bacterial cell wall in a way that's completely different than how antibiotics work? And thirdly, what I'll focus on today is actual phage therapy, and that is the use of the bacteriophages themselves to treat antibacterial infections in a way that's specific, in a way that's not targeting all of the good bacteria that are present in your body, and in a way that is self-amplifying, so the basic property of how phages work. So we need the right phage for the wrong bacterium, wrong being pathogens. And in Belgium, there are now about 50 bacteriophages that have an actual passport, a passport that's issued by a government institute that requests that specific properties are being defined. For example, this phage kills 86% of the hospital bacteria, MRSA, which you may have heard about. So if you take 100 isolates, this one phage can target about 86% of those. For Pseudomonas aeruginosa, which is another dangerous pathogen, it recognizes the specific elements on the bacterial cell wall to inject its DNA. And in this way, we can create combinations of phages or phages that are specifically tailored to this one person who has a certain infection. We collect from that person the bacteria that is causing the problem, and a personalized cocktail will be made to treat that patient. Yeah. So this is possible here in Belgium through what's called legislation called magistral preparation. This is legislation that has been there for a long time. Let's say if you are allergic to one compound in a medicine, then a doctor can instruct a pharmacist and say, okay, we need a medicine for this patient, but she's allergic to compound X. So the pharmacist is instructed to make something to avoid the thing you're allergic to, but still contains the active pharmaceutical ingredients you need as a medicine. So this exists for a long time. The new thing is that now in Belgium, phages are tentatively accepted as an active pharmaceutical ingredient. And that means that based on well-characterized phages that are in a bank that have a passport, this can be produced in specialized labs according to a monograph, and then also the quality is checked. This will go to the hospital pharmacy, and a doctor, upon prescription, can instruct the pharmacist to prepare this as a hydrogel or as an inhaler to give to an individual patient. Yeah. And this is a very interesting approach because these bacteriophages are so highly specific. This is not an off-the-shelf solution that you can just apply to tens of thousands of people. It needs to be a little bit tailored and adapted. And that's on one hand the strength of bacteriophages, but on the other hand also the weakness. Before this, we were relying on Declaration of Helsinki and compassionate use, meaning that if there is a patient in dire need, a doctor can do anything to save their life if the patient gives consent. And in the past, patients have been treated under this condition, but now with the magistral preparation, we can accelerate and treat more people. And driven by my colleague Jean-Paul Pirnet of the Queen Astrid Military Hospital here in Brussels, he has been the driving force to put forward phage therapy for human applications together with his colleagues. And last year we published on phage therapy on the first 100 consecutive cases, so no clinical trial. We're actually up to 200 consecutive cases that were primed or initiated from here in Belgium, but also exported to other countries where, based on this compassionate use, phages could be used for individual patients. Yeah, this is not always successful. You see Spain there as well, and how phages was able to help a patient with lung transplants that weren't taking due to a Pseudomonas infection. And in this way, that we're able to give a life-saving procedure. Yeah. I want to warn you, the next slides will be more graphic. I don't know if there were people here in 2016 here in Brussels on March 22nd. So this was the day of the bombing at the Brussels airport. And there was someone who was standing quite close to the bombers and basically took the full brunt of the first explosion. What you see on the right is actually what's supposed to have been the inner thigh. Yeah, so this is completely gone. She lost part of her abdomen. And the way she was brought in, she was stabilized and before taking to the hospital here in Elsene, I believe. There is no way you can do this in any sterile way. I hope you will agree this is a pure war wound, and you can say nothing else about it. So over a period of 700 days, 700 days, so close to 2 years. She was hospitalized. She had over 70 operations, of which 7 major operations. During this entire period, she was on cocktails of antibiotics, which had no effect because at any moments there was Klebsiella pneumoniae, the chewing gum bacteria that I showed you at the beginning, that was constitutively present. Yeah. So this was 2016. We didn't have magistral preparation yet, but our colleagues in Brussels prepared a phage that we analyzed and characterized and received approval from the ethical committee to actually use this. But at the time, there was no consensus from the surgeons to use it. I mean, this is a virus you're using to treat a bacterial infection. So a lot of questions were about it. And actually only after 700 days and her insistent, she received a dose of 7 days of phage therapy. And that turned things around. That coincided with another surgery and a bone transplant. And this resulted in the fact that she became Klebsiella free, the bacteria was gone. And that also allowed the removal of her fixator. So she continues her recovery until today, but I had the pleasure of actually meeting her last week, and she is doing really well. So overall, if we look at the 100 cases, because phages are so specific, we are looking primarily at Staph aureus and Pseudomonas aeruginosa. Unfortunately, E. coli, only 2 cases in 100. So we're certainly not covering everything. And that's a key limitation of phages, yeah, that we have to be so specific, so specialized. But these 100 cases, and I've given you one example, but most of the times these have been attempts at life-saving treatments. And overall, statistically, we saw a clinical improvement in 77% of cases, but this is, let's say, a subjective term because the surgeon defines what is an improvement. But we also saw in 61% of these cases that actually the infecting bacteria could be eradicated, and that's a very objective term. I told before the lecture that I was also a fan of antibiotics. I remain that because it's also shown that if you combine antibiotics with phages, your chances of success are also higher, and we have data to support this. Yeah. So how does that look like in Belgium? On one hand, we have the phage characterization and finding the right phages. We need to produce them, which is in the Queen Astrid Military Hospital. And then as a government institute, Cienzano will issue the passports and do the quality control of individual batches. On the other side, treating surgeons can approach our surgeons at the UZ Leuven, for example, for specific infections. If the right phages are present, this can be, and if there is no alternative treatment, the committee can decide whether a patient is eligible to be treated. And in that case, the pharmacist goes to work to prepare the phages, treat, and then we come back in to analyze why is it working or why is it not working. Yeah. So this is a very nice chain that we've built, but it's also very fragile. This is depending on grants. This is depending on postdoc. This is depending the person in the lab here producing not getting sick. So it's a very fragile chain that we have built, but it's been able to help so far in showing that phages can be a nice adjunct approach to treat severe chronic infections, because we need time to see which bacteria is present, culture, see if the phage is susceptible. So you cannot do that for an acute infection. Yeah, but we need more well-designed trials. But since we already are, and our surgeons have the feeling on how to work with phages, how they should be administered, yeah, hopefully that will also teach how we should do these trials. Yeah, it's all good, not all good news, because of all the patients that request phage therapy, actually only 8% get phage therapy. Yeah, in a lot of cases, because phages are so specific, we do not cover. We don't have the right phages, or there is a standard therapeutic option that's available. But I think in general, yeah, the challenges will be there to expand the phage bank and to think about sustainable solutions for cost efficient and cost of treatment, because now the patients here in Belgium, they pay nothing. Queen Astrid pays for the production. The clinics pay for the surgery. We do the microbiome analysis, all on grants that we are able to receive. Yeah, and I don't want to show this as a miracle solution. Yeah, it is going to be in the big struggle of antimicrobial resistance. Phages and other phage-derived strategies will hopefully become available, but all will represent only a piece of the puzzle to the AMR crisis, because they are so specific, and the new generation of all antimicrobials will try to maintain your healthy bacteria, the commensal bacteria that are present, and specifically target. So, but that will mean where antibiotics were able to treat all infections, let's say, as one molecule, yeah, we're now going to have a tapestry of new antibacterial strategies that are going to replace that mono approach that we have from antibiotics. But also in this way, we can curb the resistance development against all of those other strategies. I've already thanked the people from the Queen Astrid and showed them. I also want to thank my own research group, and thank you for your attention.
Okay, so thank you both for the wonderful talks. Now we have time for some questions. Maybe I take this with me here, or just I can just stand here. Yeah,
We can.
yeah. Maybe I can start just to break the ice. Just triggered for this last talk. Have phage resistance emerged so far or no?
Yes, this.
Sorry.
This occurs very it's an excellent question. This recurs very frequently. So selecting the right phages that are less prone to resistance, or I told you before that they are recognizing key elements on the bacteria. If you imagine your bacteria as a lion and it has sharp teeth, well, the phages are attaching to the teeth of the bacteria. And in order to become resistant, the bacterial lion has to lose its teeth. And in this way, if there is resistance against the phage, we try to use phages in a way that the bacteria become less virulent, become more susceptible to antibiotics. And that's why hopefully the combination will work well.
Mm-hmm. Okay, okay, I see hands already, so maybe we can pass. See,
Yeah. It's related to the question that Jorge has done, but more related to the crosstalk. So you have commented that the phages are, the bacteriophages are very specific, but there is any percentage of crosstalk with another species of bacteria? Because indeed, maybe Pseudomonas, the general or a specific species. And later, on top of that, how possible is that we face a similar situation that we are facing with the antibiotic resistance, that this phage can be spread? So, and identify another species that should not be considered as a pathogen? This one question, and later as a second question, is related to what you have mentioned about the different patients. The patient that can request the bacteriophage treatment are mainly for compasses, or there are another kind of patients that can have access? I don't know how is the legislation, which is the reaching of the legislation for that part.
The first question, I'm just a man, so I want the crosstalk, yes. Well, phages are indeed very specific. Usually they recognize only a subgroup within one species. So Pseudomonas aeruginosa phage will not infect Pseudomonas putida, which is a commensal. Yeah. So usually it's highly specific. I think so. There is not really broad host range phages that I'm aware of. And the second question, other patients.
Is it only for like extreme
yes.
cases? Or?
So yeah, there are two different approaches. The Queen Astrid will accept requests from patients themselves and will try to find solutions for any problem that they have. But this is swamping them logistically with requests. Even if there is a Dutch documentary, the week after they are flooded with requests. At UZ Leuven, we are much more stringent, my colleagues there. There we have protocols for specific diseases, lung infection, fracture-related infections, a skin disease, to name a few. They only take requests from treating surgeons, so they have clear access to the dossier. And also, and that's a sort of agreement with the Federal Agency for Health, that only in cases where phages are available, and there is no, if this is a referral case, if not all standards of care options have been used, standard of care should be used. But if these have been exhausted, then you become eligible for phage therapy.
Yes, I have a question for each of you, if you don't mind. So I'll post both, and then you can decide. Carmen, you mentioned before the talk actually about the sources of the infection that are actually very difficult to find out where you can get those antibiotic resistance bacteria. In the two cases that I know of friends that had these weird, difficult situations of flesh-eating bacteria, for example, they didn't know where they got it. And before you mentioned that the source could be different, right? Yes, so I would like you to elaborate a little on that if you don't mind. And then for Rob, I have a question, or I used to have a project called Divine, where they were working with nanofittings. Like very tiny antibodies, right? And I was thinking, could you use those kind of molecules or something similar, like an adapter, so that you have your phage, bacteriophage, and then you introduce, it's easier to randomize and prepare thousands of different adapters, and then you see what I mean? Yeah, got it. Thank you.
I answer the first question. Sometimes, very, very frequently, the bacteria that cause infection are our own bacteria that belong to our skin, our intestine, that if the defense, the person is immunocompromised, or the barrier are broken, can arrive to a place that is not the natural. So a bacteria that is a good guy that we have in our intestine can pass through the urinary tract and cause an infection in the urinary tract, or can cause bacteremia, or even a good guy that is very good in our intestine, but outside of the intestine or the skin is not so good. So depending on the type of bacteria, the origin could be different. But in many occasions are our own bacteria, that the one that are causing, because our defense, the barrier, are decreased.
Or even in your garden, if you're cutting roses and so forth, yeah, that can happen. So a very good suggestion. There are actually a lot of research groups working on engineering bacteriophages and changing the attachment sites. Even I know of some antibodies being used, but this is not something that makes it broad scale. You have to realize that this hijacking mechanism, so recognition is the first step, but also the hijacking mechanism and to proliferate within the bacterial cell, the genetic material of those phages is also fully adapted to a certain host. So it's not only limited to recognition, but also an efficient amplification within that bacterial system.
Thank you both for the presentation. It was very inspiring, very clear. And I had two questions. The first one is to Carmen. Like you mentioned very quickly that you use metagenomics to analyze also those genes related to MR, sorry. And some of them are present in plasmids, but plasmids many times can be in very low quantities when you analyze such a high quantity of DNA that is on the environment. What is the approach that you usually use for detecting them in such a complex matrix?
Yeah.
And I will ask the second question later.
Yeah, the plasmid normally we analyze through the bacteria that we obtain the genome, and we do whole genome sequencing, long read, for example, by whole genome sequencing, and in this way we can do the cartography of the plasmid, and we know exactly where they are, because otherwise in metagenomics it's more complicated. So we have, for example, I didn't explain, but in the store we have analyzed all the plasmid they carry, and we have compared with the genome and plasmid of humans, animal, that the genome that are deposited in gene bank databases, to see if along the migratory route we can get any comparison, any correlation. So we have performed, I think, one of the biggest studies in wild migratory bird in relation with this specific resistance we are working. But we do through whole genome sequencing of bacteria that are isolated, and we can do the cartography of the plasmid.
Okay, thank you. And the second question is for Rob, and it's about phages. I'm curious about the administrative dose and the dose, because you mentioned that sometimes there are aerosols. So when I think in antibiotics, we basically can take a pill and we can get cured in most of the cases like that. But with phages, can they actually pass the gastrointestinal tract and go to blood, or does it have to be localized?
Good question. First, I want to make a small note that I didn't mention. Other dear colleagues are at the Eliava Institute in Georgia, where they have decade-long experience with phage therapy application. And a lot of the protocols and how much phages that we provide is also based on their practical experience that they have in Eastern Europe in applying phages. It's correct. So applying phages is a delicate matter. If we're talking about fracture-related infections, this is usually done locally during the surgery while the patient is open by the trauma surgeon. There we have the best results. In lungs, it can be more challenging. In gut, it can be more challenging because of the complex microbiological environments. Indeed, passing through the stomach can be an issue. Some phages are stable to a pH of 2. Others are not, but if you combine it with antacids, for example, you can temporarily increase the pH and allow a better transit to the gastrointestinal system.
And just last question, promise. On this, viruses in general have a high rate of mutation. So you can imagine that the virus you apply, the phage you apply, will mutate soon. So how do you deal with this when you're applying a treatment?
That's a very good question. Actually, we try to use that. Sometimes patients will have a bacterial strain, and we have phages that are sort of infecting, but not efficiently. We will then train the phage and mutate the phage so it becomes more adapted and will be more infecting that specific bacteria. So we're actually using mutations to our advantage. If you imagine these phages and bacteria, they're in a continuous arms race where bacteria becomes resistant, mutations from the phage accumulate. And we consider this one of the strengths that bacteriophages have in combating. So we're actually using that. But on the legislative point of view, this is a key problem because you will see in many European countries that you can only have a medicine that's stable, that cannot change, that has undergone GMP production in a very fixed way. And yeah, this is simply not compatible with this personalized approach. And you see in a lot of countries, including Spain, unfortunately, that are really hanging on to this rigorous framework, not allowing changes to occur to actually optimally use phage therapy. So currently in Europe, this magistral approach, which doesn't require GMP, is present in Belgium and has recently been allowed in Portugal. And I would argue to this crowd, if the Portuguese can do it, so can the Spanish. I don't know if there's legislators here or of people of political influence.
So thank you for the nice talk. Just a practical question for the phage part. So it's known that the use of antibiotics in the animal farming is one of the main causes of antibiotic resistance. Do you think that bacteriophage can be an alternative, in the future for this kind of large-scale treatment?
In animals?
In animals, yes.
Yeah. I'm going to give you my personal opinion. I hope they don't start using it in animal production for all of the reasons that Carmen has outlined. For me, phages are a valuable resource that we can use. The key thing will also be cost. You need to be very cost efficient, and when treating humans, that's less of an issue. But in animals, every euro cent counts in the animal feed. Where what I do believe in is applications in crop protection, where if there are crops that are threatened by specific bacteria that reduce the harvest. But there, phages don't have to contend with an active immune system that's present in animals and humans that can also limit or eradicate some bacteriophages that are inside of the body.
Thank you.
Questions? Maybe on the back first.
Thank you. So the question is about the One Health system. So my question would be more or less if it would be difficult to use this antibiotic resistance virus or diseases to be used to weaponize, for example, to damage a population on purpose, or it just appear like that.
So do you think bioterrorism, AMR, could be used for bioterrorism?
Of course, can be used mostly if antimicrobial resistance is in a pathogenic bacteria. It is in a commensal bacteria is very difficult because commensal bacteria can disseminate without causing, but are very important for the evolution. But for bioterrorism, it has to be in pathogenic bacteria. I don't imagine put in a specific in E. coli because it will take some time just to get the, but if you put in another bacteria, perhaps. Yeah.
But maybe to add to this, I think in general, viruses are much more dangerous in that regard. For bacteria, some patients need to be immunocompromised, so you will not have the same level of spread like you do with the COVID virus. Yeah, a COVID virus, yeah, that level of spread you will not attain with bacteria. Yeah, you can think of leprosy or maybe as a most extreme case, but yeah, usually hygienic treatments or measures can limit this much better than in viruses.
Another question here.
Is the climate change on the equilibrium of
Yeah.
the viral and bacterial population?
Yeah, climate change is affecting to antimicrobial resistance. We have seen that, for example, migratory birds, due to climate change, they are changing their habit. And instead of migrating, they are maintaining in landfill, eating in a landfill for a long time. So we have analyzed, for example, a store that are feeding in landfill, and the resistances that we detect in this store feeding in landfill is very high. So climate change, of course, is affecting antimicrobial resistance. This is an example, but there are many other examples of this correlation. And of course, in the dissemination of a specific pathogen and so on, but also in antimicrobial resistance, mostly because are changing the habits of some animal or, yeah.
I'm still curious about the phages. So the question is, do you know any secondary effects, any drawbacks of using phage therapy? Because it sounds really great.
Yeah.
But
So for the 100 cases, we haven't seen any serious adverse effects that would directly impact the patients. It is seen that phages can have an immunomodulatory effect, but that's not necessarily negative, quite the contrary. So no, but again, the main drawback is the specificity, the needs to identify in advance your pathogen. Is it susceptible to the phage? And again, our own immune system can maybe neutralize phages if it's in the bloodstream through immunological response. So there, I hope my message was not too rosy. We have to be careful and precise. It's not a magical solution.
and what is the prevalence of these phages concerning type of bacteria? Do you find more easily phages against gram-negative than gram-positive, or?
Yes, definitely. It's harder to find a phage against Staph aureus. There are also phages that are not all directly killing the bacteria, but can, for the people who know lysogeny, who can go in a symbiotic interaction with bacteria. Those are undesired. But yeah, so others are more easy to find, like Pseudomonas phages, you will find easily. No one has ever isolated, to my knowledge, any stable Legionella phage, Legionnaire's disease, but for Legionella, I've never seen any phage. And I think the more weird your bacteria is, the harder or the more hard it is to grow in the lab. It will also be harder to find and produce phages in a good way.
Hello, good afternoon. Thank you very much for the presentation. Just two very quick questions. The first one is related to the use. Is there any kind of work on a diagnostic test to identify the use of phage? This is the first question. And the second one related to the clinical trials. I understand that with the compassionate use, it's very difficult to have clinical trials, but do you have something in this way, something similar to this? Thank you.
So yes, in terms of diagnostics within Europe, I don't know if you're a specialist, but UCAST, who is responsible for diagnostics, there is a special working group to make consistent the way phage testing and phage diagnostics occurs. There have also been biotechnological developments using phages or parts of phages to have very sensitive detection of bacteria. In terms of clinical trials, Horizon Europe has, and that call just ended, there was a call specifically for clinical phase 1 and 2 trials using phages for a randomized blinded clinical trial. So I heard there was a lot of enthusiasm to and a lot of consortia which have entered this call. So hopefully in the future we will see clinical trials happening. There have been in the past, but these have been these fixed cocktails that are not specific, not tailor-made, and those trials have failed. So we need to be very careful. This will be, I'm going to say, the last chance to get it right, I think, within Europe to put forward phages as an alternative.
We have time for maybe one last burning question. Elena is raising her hand. So
I will take advantage that I hold the mic. So basically it's the same question for both of you, but I really like the metaphor that you use about the puzzle, right? Like the phages are just one piece of the puzzle to fight AMR, but also like the One Health, like how do you see, you have to see it as a whole. So what would it be the pieces that you think we should focus, or well, you should focus, well, you will focus in the future, I guess, each of you, or what do you think is somewhere where all the, where this policy science people should be trying to focus right now, or so?
From the One Health perspective, it is very important to take into account the environment. The environment is a forgotten ecosystem that is very, very relevant. So I think now there is a big project in Europe that gather all countries. We are participating in this project, the University of La Rioja, that try to see all the sector, humans, animal, and the environment, because the environment in most of the European countries is almost not studied, is what we have been detecting. And for example, the wastewater is a very hot spot that has to be treated. So we have to understand, we have now new tools, new genomic and metagenomic tool to understand the flows and to understand which are the forces that drive the evolution of resistance and the way of transmission. And of course, alternative to antibiotic, that is also a very, very relevant topic that we have to do. And also, other things that are very important, preventive strategies, is not only to develop new antimicrobial, new therapies. We have to work in new preventive strategies. We have to see how to use better what we have. That perhaps is important to do a good analysis in order to improve all we have, and of course, to do research in new things.
I completely agree with that, but I will take it in a different direction. I think politically something completely different is necessary, and that is investments. I think, I don't know, the last time you went to the pharmacy and had to buy antibiotics, they will not have cost you more than a few euros. Yeah. So antibiotics are not profitable to companies. An antibiotic treatment is 1 or 2 weeks. This is very different from a statin for your heart, which you will take for the rest of your life by the same company. So there is not a lot of industry drivers and viable business models, and it's and that's, sorry to be negative, but that's one of the big concerns that I see, and that the companies are actually withdrawing from developing new antibacterials and despite the societal need, because simply the business model isn't there. I think that's one element. And secondly, I'm a believer of a tapestry of we need a new generations of antibiotics. We need alternative strategies that might be based on phages. They could be other proteins and enzymes. I don't know if you have flamazine in your fridge. This also contains enzymes which have antibacterial effect. So more of these enzymes, other molecules called bacterocins, which are basically peptides made by one bacteria to another. All of these things, and also including probiotics, so good bacteria. Once we eradicate the bad ones on the wounds, that good bacteria take over that niche. So these are all very concrete strategies that need to be integrated and combined or used in specific areas.
Thank you.
Okay, we don't have time for more questions. But now we will have a reception with some Spanish tapas that you're all waiting for. But I want to thank the speakers because they are taking their time to be here and to communicate about this very important topic. So I really thank you for investing your time in coming here and giving these fantastic talks. I also want to thank all the people that came, because you're also coming here in your free time, and you're genuinely interested in this thing, which we thought it would be important to also communicate and talk about. So thank you for coming here. If you think that these events that we organize have things to improve, don't tell us. No, tell us, because we will be willing to improve the way we do things. And again, thank you for Instituto Cervantes for hosting our events, and also the funding institutions, the Embajada de Cultura and La Fundación Ramón Areces. And okay, now let's go eat and drink. Thank you.
A big applause.